Thank you for the kind introduction, Chris. So if you want to study the plant-animal interactions of carnivorous plants, we can do this also in dried and living collections, and I want to show you some examples for these. Carnivorous plants evolved in about 860 different species with the various trap types comprising sticky traps or the very advanced suction traps, the rather straightforward pitcher traps, and a lot of different diversity. And in order to study their prey spectra, like in the famous Venus flytrap, or in Sarracenia minor, a North American pitcher plant, which is also featured on the program of this symposium. Normally, to study the kind of prey they capture, you would have to study these plants in the wild. And if we do these field studies in their natural habitat, we see that even if they display the same trapping type, like here the sticky traps in sundews, sometimes their prey spectra are very different. There are species which capture rather large insects, such as these butterflies here in this Australian sundew species, where some specimens are literally covered in prey. And this is also obvious in herbarium specimens. Even in historic herbarium specimens, like this from Kew Herbarium collected in the 1970s, we can see here on the right-hand side different beetles, different moths, which can, by the specialists, be identified even to the insect order, or even the insects can be identified to genus or species. Here, for example, this large butterfly, which is visible on this herbarium specimen. But of course, these only represent single snapshots of the prey spectra of this species. So usually we need to study the natural populations to get an idea of the prey spectra of carnivorous plant species. So how is this best done? Because some of them capture rather small insects in high numbers, where others have rather large prey items, but in smaller numbers. So the question arises about prey attraction, prey spectra, and potential prey selectivity. So I have to show you a little part of my research, which is done in fieldwork, and the basis of this, which I did using living collections and herbarium material. So first, for the fieldwork, our study system was a group of sundew species, closely related sundew species from tropical northern Australia, which look very similar because they're closely related. Here, just two examples with different flower color. And the species on the right-hand side has a sweet honey-like scent on the leaf, so it has odorous leaves, whereas the one on the right has no trap scent. And in order to compare prey spectra of different species, we developed a method which is done in situ, so in the natural habitat. We selected the 5 youngest leaves of the active trap leaves of each specimen of 10 individuals per population. And we photographed every little prey item with high resolution macro photographs. So every little thing, insect, which is captured on these leaves. So we made some close-up photographs to identify these different insects by photography. And then we sampled these leaves. So we collected these 5 leaves and put them in alcohol for DNA barcoding in the lab. So we extracted the DNA, the genetic material of the insects, and were able to identify these insects by genetic markers. So this enables us to tell something about the prey quality of each species. So what does each species capture? And this is really a very smart method using this DNA metabarcoding, because then we get per plant sample an idea, a percentage of how many DNA barcodes of each individual prey item are found. So we can trace back here, for example, this moth genus Uthathaisa. These 2 large individuals were represented by 38% of the barcode reads. And so we can identify each prey item sometimes to insect genus or even to insect species without being an insect taxonomist. And we also get new occurrence records here for Northern Australia, for example, where these insects were not recorded previously, but they are now recorded thanks to being captured by these plants. And what is of interest for our research is the prey quality of sympatric species, like here this red sundew, and it's closely related. There's a different species, this yellowish-green species, and this is indeed species dependent. By using these DNA barcodes, we can now compare different species, and for example, we see that this is the red species here, Drosophila. It captures a certain percentage of Orthoptera, so grasshoppers, which the other species did not capture. And so we find also for our example with the fragrant and the non-fragrant species that, for example, they differ in the number of butterflies or Hymenoptera, so insects which can sensor the honey scent, so nectar-feeding insects, which are by a much higher percentage captured by the fragrant species. So these carnivorous plants quite often are something like a selective flypaper, so they do not capture everything that is flying or crawling around, but it seems that at least in species which co-occur in the same habitat, that there's a niche separation and the different species capture different insects. However, in order to test this method, we need to know, does the state of digestion or the age of the captured prey play any role on the DNA barcoding detection? Because our method, which we use now for these field collected samples, strongly depends on that we detect everything which is captured by these plants, at least on these 5 youngest leaves selected. And to study the influence of prey digestion, of these digestive enzymes digesting the prey on the detection of the prey in the DNA barcodes, that's where living collections then come at hand. Having a large sundew collection that gives us a lot of study material in the greenhouse where we can make digestion experiments and compare these with the DNA barcodes. We did this in Munich Botanic Garden. So this label which the gardeners, the greenhouse keepers kindly left with the plants we were studying says research. So please do not water from above, spray, etc. So what we did, we were using different carnivorous plant species representing different trap types. And the good thing in cultivated specimens, so these 3 or 4 individuals of this sundew, Drosera capensis, the Cape sundew, they are a clone in cultivation. So they're genetically identical. So we assume that what happens on this leaf here, for example, regarding prey digestion, is identical than on that leaf here, because we were always using leaves of approximately the same age or developmental stage. So what we did, we fed these plants. And we did not just randomly fed them. So what they had for dinner or lunch, it was a defined menu. So what was on the menu? We were using insects which were readily available in pet shops as food for reptiles or fish, like here these midges, 2 different species of fruit fly, Drosophila melanogaster, a small fruit fly, and larger fruit fly species, small crickets, springtails, and bean weevils. So 6 different insects. And why these very different insects? To see if we can detect them all in the DNA barcoding. And we were using insects with very different morphology. So some small insects like these springtails, or soft-bodied insects like these midges, or on the other hand, very hard-bodied insects like these weevil beetles, where we assume that prey digestion in some of them is faster and in others slower, and maybe not all parts can be digested. And then what we did, we were using these insects in frozen state. So we were killing them by deep freezing them, just to avoid that during our experiments and some of these replicates, the insects would just, free themselves and disappear. We were applying these insects to the leaves. We were using 5 replicates of every digestion step, and then we were sampling these leaves. So This is what happens on these leaves just after application. So the tentacles of these sundews start to bend over. The plant recognizes this food, even if it's not moving anymore. It recognizes the proteins. And then after about 2 days, the leaves completely unfold and prey digestion is already going on. So the plant enzymatically breaks down the proteins and takes up the nutrients from this prey. And after about 1 week, the leaves start to open again, and what is left on the leaves are the remnants, the chitin exoskeletons of the insects, which cannot be digested. Now, in order to infer the role of prey digestion or the influence of prey digestion on the detection in the DNA barcodes, we sampled individual leaves, 5 replicates from each individual after fresh application of the insects, then after 1 day, after 2 days, and so on, until the digestion ended after 7 days. We did this in different trap types. In pitcher plants, for example, there's nothing to show in the picture. It looks identical between freshly applied prey, and then we sampled them also in individual steps. And here the time span was much longer because these pitchers last for quite long. So we sampled them. The last sampling was after 1 month. In the Venus flytrap, we did the same. Here it was a bit difficult because the plant reacts to moving prey. So we only applied 4 of these insects, and the last one, these beetles, we applied as living insects, so let them crawl around so that the trap's closed. And then again, we sampled these individual traps of the plant after a certain given time span. Here, for example, while prey digestion is fully going on, but also after these traps reopened after 5 days, where you can see here the remainings, the undigestible items here, if there's some still DNA to detect. We used also different species of sundews, tropical and temperate species, to see if the temperature or also the phylogeny, so the affinity of the species, has any influence. And again, we sampled this here. You can see how the leaves curl around the prey during digestion. And this is also visible then in the sampled leaves. We sampled them in alcohol. That's how DNA barcoding starts. And we did this until the very end, until the leaves opened again, and also the leaves already showed first signs of degradation, just to see if we can still detect some DNA. In the pitcher plants, this is also very showy because the freshly opened pitchers, you can still identify these insects. Here you can see the cricket, for example, and the beetle. Also the fruit flies can clearly be identified, while after 1 month, this is just a crumb of fungal hyphae and algae and heavily digested prey items. The results are not fully analyzed yet, but it's really amazing how sensitive this DNA barcoding is, and it seems that at least some legs of these insects are not digested and are not degraded, and we have traces of all the prey items even after 30 days in these heavily digested items in the pitcher plants. So it's really like a reconstruction of a crime scene. We can see what the plant has eaten because apparently every insect seemed to leave some DNA traces on the plant. A different crime scene of plant-animal interaction in carnivorous plants are kleptoparasites like this ant stealing prey from the sundew, and herbivores, so insects feeding on carnivorous plants. For example, a famous kleptoparasite in Australia are these sundew bugs, these assassin bugs, which live on Drosera and which feed on these insects. And even in the DNA barcoding, which we did in the field, we can detect these bugs even if they are not there, because apparently they leave some traces of DNA while sucking on these insects. And one insect which interests me quite a lot is a herbivore of Drosera. These are the sundew plume moths, Buclaria. There's one species, Buclaria paludum, which occurs in the entire Europe, also in the UK, and it was once considered to be the rarest moth species of the United Kingdom until people started to look at the habitat of the larval food of this moth. And this moth is monophagous, so the larvae feed exclusively on a single plant species and this or plant genus, and these are sundews. So this little caterpillar here lives in probably the most dangerous habitat for an insect on a carnivorous plant. However, it's not eaten by the plant because it feeds the carnivorous structures. So it feeds the tentacles, these carnivorous glands of the plants, and so it's not eaten up by the plant. You can see it here feeding on these glands. The leaves leaving some traces of frass on the leaf, so it leaves a very characteristic feeding damage on the leaves. And the later instars of these caterpillars even feed on the prey, so they become carnivorous caterpillars, and they feed on the captured prey. And then they pupate on the back of the leaves, so on the non-dangerous part, and then finally the adult moths hatch. And the very interesting thing is that both feeding damage, which is very characteristic, these tentacles being eaten up and the frass, and also the caterpillars and the pupae can be found on herbarium specimens. Here, for example, you can see this when the plant specimen here was pressed, this caterpillar was also pressed, and it's clearly Buchlaria. It's very unique. The leaves also show this feeding damage. Or on this Drosera species, even two of these pupa can be found. So you can see here one of these very characteristic pupa, and this is Buchlaria. We also evidence this then by DNA barcoding that this is Buchlaria. And this is very interesting because if we look at the distribution map of Drosera, here the more red it becomes, the more species. The diversity centers of Drosera are on the southern hemisphere in central Brazil, in the Cape area, and especially in Australia. And where Buchlaria has been recorded so far, the genus currently has 7 to 9 species, depending on the moth specialist. There's one species in northern hemisphere of Europe and Asia. There's one species in North America, one species endemic to Brazil, 3 species in Africa. And the interesting thing, I was wondering, why isn't there any Buchlaria species in Australia? Because it's the center of diversity of Drosera. So I almost predicted that Buchlaria, the sundew plume moth, has to occur in Australia. And indeed, I found them while traveling in northern Australia, feeding here on the Drosera. Clearly, these little caterpillars belonging to Buchlaria. And interesting thing is then we were able to prepare a distribution map of this moth without traveling there because of the herbarium specimens, because of the pupae or the caterpillars pressed with the Drosera herbarium specimens. And with this Buchlaria moth, meanwhile, we have more records, distribution records of this moth worldwide coming from Drosera herbarium specimens, which I examined, than from moth specialists looking at collected specimens of the adult moth. So this is clearly under collected, this moth. Probably here it's the botanists which contributed more to the distribution pattern of, or the distribution records of this moth species. Because this moth, like the prey captured by the sundews, comes as a bycatch on these herbarium specimens. So studying herbarium specimens of carnivorous plants can give us some hint on the ecology on the plant-animal interaction or actions of these species. And one interesting fact is that the first ever published mention of plant carnivory is also found on the herbarium specimen. It's not in a published book, but it's in a herbarium specimen. When we think of who, first evidenced plant carnivory, we always think of Charles Darwin, who published this benchmark book in 1875, Insectivorous Plants. And indeed, he widely published this, or we made widely public this phenomenon of carnivorous plants. However, he was not the first one to discover this. This was almost 100 years earlier, the German botanist Albrecht Wilhelm Roth, who wrote a little publication on sundew species. and the 3 European sundew species. And in this, he wrote that the enfolded leaf, which keeps captured an insect, seems not to open again until the caught insect has decayed. Yet I believe that the habit and character of these plants aim at obtaining their nourishment for preservation and reproduction. He published this in 1782. So clearly evidencing that or theorizing that these are carnivorous plants. So this was almost 100 years before Charles Darwin published then his famous book, Insectivorous Plants. And interestingly, the first mention we have of Roth on this theory comes from a herbarium specimen, which is found in Munich Herbarium. And here, this little envelope, this little sheet where the specimens are hidden inside, has this little note, nota bene, folia inflexa tenent insecta mortua. So Latin for note, the incurved leaves hold dead insects. So he clearly wrote this on the herbarium specimen he collected. He wrote an ecological observation he made, probably on the living plant, of course, before he collected them, but he communicated this to his contemporary colleagues, botanists, by this herbarium specimen. And that he really wanted to communicate with these specimens, he mentions in a different publication in 1800 in his Catalecta Botanica, where he mentions in Latin that he cum specimenibus communicavit, and that his observation he has communicated with specimens. So Roth was aware of one function of herbarium specimens, namely not only to that this herbarium specimen preserves the plant and entire morphology, but also serves as scientific as an example of scientific communication to other people. And with this, I'd like to end my presentation. And thank you for your attention. And I'm open for questions at the end of this session.